So it's a pleasure to be here tonight. I want to thank Caroline and the LPC for organizing this talk and for inviting me to be here tonight. And I also want to thank you all for chiming in and for your interest in malaria and in common loons. So my name is Ellen Martinsen, and I am a researcher in the malaria lab in the Department of Biology at the University of Vermont. I'm also a research associate at the Smithsonian Conservation Biology Institute. And the work I'm going to be presenting tonight is completed in just a very small part by myself. There's a huge team of collaborators involved in the work and research that I'm going to be sharing with you tonight. Some of them are listed here. Most of them are listed here. Some of them are pictured here. The Loon Preservation Committee has been a huge part of the loon malaria work. They've been sharing blood samples with me for over a decade, and this work would not be possible without the LPC. It also wouldn't be possible without the key people pictured here, and you'll be hearing about some of these people as we head through the talk. So common loons are experiencing population declines across their breeding range. There's a beautiful paper that was just published in December of 2020 in Ornithological Applications, and this was an eloquent study because it involved studying loon populations over a course of 27 years. So this is a long-term study and took place in Wisconsin and was headed up by Walter Piper and colleagues. And what they found over 27 years of studying banded loons is that chick mass has fallen by 11%, mortality among young chicks has increased by 31%, mortality among old chicks has increased by 82%, fledgling success has decreased by 26%. Return rate of non-breeders or floaters, these are the birds that are floating around looking for a breeding lake or pond, that their population, their return rate has plunged by 53%, and the adult population has declined by 22%. So this is pretty surprising, and these are data that only emerge after a long-term study. And modeling the data from this study, they estimate or predict a population decline of 52% by 2031. This is pretty remarkable, given that we thought that loons were on the recovery and that they were doing okay. And it also suggests that we need to pay closer attention to what's going on with our common loon populations. There are a number of factors, mostly if not all anthropogenic, that are going on to drive these population declines in common loons. But something that hasn't been yet looked at is what is the role of emerging infectious diseases? And so this is where the common loon malaria parasite study comes into play. So I always like to start with this slide talking about malaria parasites. So malaria parasites are the critters that cause the disease malaria. And when I'm talking about malaria parasites, I'm talking about an organism in the kingdom Protista. So these are single-celled eukaryotic organisms. They're protists. They have cell membranes, they have a nucleus and a mitochondria, and they are specifically in the genus Plasmodium. So for the remainder of my talk, when I refer to malaria parasite, you'll hear me say it over 100 times. I'm referring to parasites only in the genus Plasmodium. And you're probably familiar with malaria parasites in humans. Here's the life stage, the life history stages or the life cycle of malaria parasites in a human host, and it's similar in wildlife hosts as well. And what's so cool about these parasites is that they have life history stages not just in a vertebrate host, but also in a vector host. And the vector for the majority of the malaria parasites are mosquitoes. So you can see there are life history stages in the vertebrate, and this includes the liver and also the blood cells, and also in the insect mosquito. And it's this mosquito host that's important, and I'll be talking about the mosquito host later on, because the mosquito is especially subjective to changes in temperature and humidity. When it comes to studying malaria parasites, it's the life history stages that are in the blood cells that are most studied by parasitologists, because you can take a drop of blood from a vertebrate host. It could be a human, it could be a common loon, and when you look through the light microscope, these are the parasite life history stages you see. And these life history stages in the red blood cells, if we follow their levels or intensity or parasitemia over time, this is the track of infection or course of infection we typically see in a co-adapted, co-evolved host parasite system. So you have the pre-patent period after the injection of mosquitoes by the mosquito into the vertebrate host. Let's say it's a common loon. You have these parasites increasing in number in the red blood cells. And then at this point, the host immune system comes into play. It kicks in the immune system. The immune system kicks in, kicks back, and decreases parasite levels. And either after that point, the parasites either are cleared from the vertebrate hosts or they remain at low levels. And typically in birds remain infected or have their infection for the course of their entirety of their life, with relapses of infection that correspond to periods of stress. In birds, it's usually around the breeding season when breeding hormones are raging in the blood and birds are undergoing increased level of stress. But again, this is the course of infection in a co-adapted host parasite system. If it's not a co-adapted host parasite system, you have an increase in virulence because the host immune system can't kick down the parasite numbers, and this leads to disease and death. And this is what we see in human malaria. So there are a handful of malaria parasite species in humans. The most virulent is Plasmodium falciparum. In this, we know this parasite happened in humans as a result of a pathogen, a parasite switch or spillover from gorillas into humans. And we know the human health implications of malaria are huge, with millions of cases worldwide and many deaths. Similarly, malaria parasites have switched into wildlife hosts and likely has caused virulence and death. So this is the story of Plasmodium relictum. Many people, when they think of the Hawaiian Islands, they think of paradise. When I think of the Hawaiian Islands, I think of extinction paradise, extinction capital of the world. And a lot of that extinction is due to Plasmodium relictum. So if you've ever been to Hawaii, you know it's inhabited by a diversity of endemic Hawaiian honey creeper species. They lived out there in isolation without pathogens for hundreds of thousands of years. Humans came along, we introduced mosquitoes. We also introduced non-native birds carrying malaria parasites. So these malaria parasites switched into the endemic Hawaiian honey creepers. And as a result, endangerment and extinction has happened. And it's pretty sad, but only 18 of the original 56 honey creeper species are still alive and present on the Hawaiian Islands. And malaria is partially to blame. Malaria parasites are wonderfully diverse and widespread. So we know they're in humans, they're in other great apes, they're also in primates, they're in rodents, they're in bats, they're in ungulates, including deer, including our own white-tailed deer, and they are in lizards and they're in birds. They're on every continent except for Antarctica, and they're more prevalent and more common around equatorial region, so tropical region, and also in temperate regions. But we also know that climate change is coming into play here, and it's shifting the distribution of many different organisms. It's shifting the distribution of birds. So just within the last 3 decades or so, we've seen a number of new bird species occur here in northern New England. So I'm in Vermont now, but in Vermont, we didn't used to have northern cardinals or eastern tufted titmice. These are all new arrivals in the last 30 years or so. So climate change is driving the northern range expansion of a number of vertebrate species. And it's also changing the range expansion of the vector or the mosquito species. So there's a recent study that showed that in Connecticut, the number of mosquito species has increased over time, as well as the abundance of different mosquito species. This is a study I was involved with a group of New Jersey vector ecologists and entomologists, and we were focused on Culex erraticus. This is a species that breeds in swampy areas around ponds and lakes. Its traditional range is South America, Central America, Southeastern United States, but over time, it's been expanding its range northward, and now just recently it was documented in Vermont and in southern Canada. What's so special about this species is what we studied. The main question was, what is Culex erraticus feeding on? So we took blood-fed mosquitoes. There's one picture here. And what we did is we sequenced vertebrate DNA from that blood meal to figure out what that mosquito had fed on. Results showed that this mosquito species loves to feed on birds, and over half of the blood meals were from medium and large-sized water birds. And it's no surprise that common loon DNA was found in a number of these mosquitoes. So we know that this mosquito species is moving northward, and we know that it likes to bite common loons. It breeds in similar habitats as common loons. We also know that it transmits malaria parasites. In terms of what is it in the question of what is it transmitting up north where it's just recently arrived is unknown. So you can see how much climate change may be impacting host parasite associations. So here's the life cycle of a mosquito species from egg to adult. I just want to show you that the development of this mosquito from egg to adult is very temperature dependent. So at 62 degrees Fahrenheit, it takes 28 days for a mosquito to go from egg to adult, whereas at 86 degrees Fahrenheit, it only takes 7 days. So these mosquito vectors are very subjective to differences in temperature. Likewise, the malaria parasites are as well, because they're developing in that mosquito host. And parasite development it takes 26 days in the mosquito host at 62 degrees Fahrenheit, whereas it is sped up to developing in just 6 days if that mosquito host is at 86 degrees Fahrenheit. So climate change is leading to differences in vector distributions and abundance, as well as the change in numbers of parasites. And indeed, this is what studies are showing. They're showing that climate change increases the risk of malaria in birds. And just recently, malaria parasite transmission was documented in Alaskan bird populations. So we know malaria parasites are on the move northward. What about common loons? So this is a species that we all know and love so much. We also know it has a very northerly distributed distribution. We also know that it's an icon of the northern wilderness. So this is a range map of common loons. Where it's common during the breeding season is hot pink, where it's rare during the breeding season is light pink, and the overwintering areas are shown in blue. So we know this bird, common loons, are up north. What do we know about their malaria parasite fauna? We know that they do not have malaria parasites. This is historically known in parasitological textbooks. The world's leading parasitologists will say loons do not have malaria parasites. We've been screening them for over almost 100 years, hundreds and hundreds of loons, and we have not found any malaria parasites. These are the studies that have been conducted so far, and I'm just going to highlight this one because it involves two of my collaborators. So this is Inga Sidor and Mark Pokras from Tufts University. They screened blood smears from common loons from the Northeast, including 114 birds from New Hampshire and Maine, and they know what they're looking at. They were looking at hundreds of thousands of blood cells, and they did not see a single malaria parasite. And these were birds sampled in the early 2000s. So again, we know that malaria parasites loons do not have malaria parasites. So this is where I came into the picture. I do PCR screening for various wildlife pathogens, and Mark was able to get me some blood samples collected by collaborators from across the Northeast. And so I conducted a common loon malaria parasite survey, again by PCR. So these are blood samples that were taken by loons when they were live captured during the breeding season for banding purposes. So that a blood sample was taken from the leg of the loon right before they were released back on their lake. And I was able to get a pretty good sample size from 3 different years across 4 states. So this is New York, Maine, New Hampshire, and Massachusetts. And this is what I found. I found that in 2010, 5.4% of common loons were infected with malaria parasites. In 2011, 19% of common loons are infected with malaria parasites. In 2014, the prevalence goes up to 38%. These are statistically significant numbers. So we have seen that malaria parasites are showing up in common loons, and they're statistically significantly elevating over time. This is pretty surprising. My mouth fell open when I did my first PCR. So just to give you an idea of what I found, this 2011, I had the most number of samples. You can see I found infected birds in all 4 states. Sample sizes are relatively small in New York and Massachusetts, but I found infections, Plasmodium infections, from all 4 states. If we just look at malaria parasite prevalence over time in Maine, I had the most number of birds from Maine per year. You can see that prevalence goes up over time. And again, this is statistically significant. So malaria is showing up in lanes and it's going up pretty quickly. Some of you may be wondering, wow, I wonder if some of the lakes around me have malaria. These are just some of the lakes from my study where I found malaria-infected common loons. Maybe some of you here tonight and live close to some of these lakes. And then what about if I look under the light microscope at blood smears from the same birds that I found to be infected by PCR? I can see malaria parasites in the blood cells, so they are there. And here is my mini-me. This is my daughter. She's 9 years old, and she's looking at a malaria parasite in a common loon blood smear, and she's excited to see the parasite, although we feel it's not so great for the loon, but when you see the parasite under the light microscope, it is a pretty amazing thing. The avian red blood cells are these oval shapes. They're nucleated, so they have that pink oval in the middle, and those little blobs with the little dots in them, those are the malaria parasites. So they are visible in the blood smears as well. To understand the diversity of these parasites, we need to use genetic methods. We can't just identify species of Plasmodium parasites by light microscopy. So I did some phylogenetic work. I found 6 different malaria parasite species in common loons from the Northeast. This was a huge surprise. I was expecting maybe 1 or 2 species, but they're infected with 6 different species from the samples that I have. And if we ask the question, what other birds are infected with these 6 Plasmodium species, they are found in various different songbirds from North America and South America. Some of these I found infected through my own previous studies. What's surprising is that 3 of these malaria parasite species, the ones that I have circled, these 3 malaria parasite species are just recently, within the last 10 years, been found in birds at more northern latitudes. These are birds in southern Quebec, in northern Quebec, in British Columbia, and Alaska. So these parasites are just for the first time being found at northern latitudes in birds. And what are the mosquito vectors for these parasites? That is the big unknown. Through my own work, I know that one of the malaria parasite species in common loons is transmitted by Culex pipiens. If anybody's familiar with mosquitoes, this is the northern house mosquito. This is also a mosquito that's not native to North America. This mosquito was accidentally introduced to North America through ship ballast water that was carrying the larvae of mosquitoes. And now this mosquito is pretty well established in North America. It's one of our most common mosquitoes. It's pretty abundant, and it's just increasing as climate change increases temperatures. So again, so much is unknown. So fortunately, in the malaria lab here at UVM, we have a graduate student. This is Alyssa Newhouse. Some of you may recognize her. She was the loon biologist for LPC last summer on Lake Umbagog. Here she's shown here holding a loon chick, and she's doing her master's research on common loon malaria parasites, and she's sampling loons from across the entire southern edge of their breeding range. So not just Northeast, but also Minnesota, Montana, British Columbia, and Washington, as well as some samples from Wyoming. And she's also screening mosquitoes to better understand what are the mosquito species that are transmitting malaria parasites to common loons. So stay tuned for an update on malaria in common loons. And importantly, also, Alyssa is screening samples provided by Collaborators from 2019 and 2020. So we'll get a better idea of just how much more infected, or how the prevalence in common loons has increased over time. So another project I was involved with is looking at the exposure of common loons with mercury. So for those of you familiar with work through the LPC and also the Biological Research Institute, they've been looking at blood mercury levels in common loons. Loons are an amazing indicator of aquatic health because they're the top of the food chain. And we know from coal-fired power plants in the Midwest, they emit mercury into clouds, which then come east, and it gets precipitated as rain into freshwater bodies. And then again, through the food chain, this mercury accumulates into predators, top predators, and common loons are one of them. And we also know that mercury is an immunosuppressant. It reduces the functioning and the capability of a host's immune system. So how does this exposure to mercury influence susceptibility to malaria parasites? This question has only been answered in lab models. There's a nice study that was done with rodents and rodent malaria, and it showed that lab mice that were exposed to mercury were more apt to be infected and less able to resist malaria parasite infection, as well as acquire immunity. So this work was done with an undergraduate here at UVM, Sarah Weiss, and also the mercury data comes from the Biological Research Institute. And the loons that we used for this data set came from across the Northeast, including New Hampshire. And this is what we found. If we look at blood mercury level and infection status, we see that the higher your blood mercury level as a common loon, the more likely you are to be infected with malaria parasites. So this is pretty surprising. This hasn't been shown in a wildlife host before. These data are preliminary, but they are significant. We'll be increasing sample sizes and increasing and doing more work on mercury. So stay tuned to that too. But we have preliminary data to suggest that mercury impairs a common loon's resistance to malaria parasite infection. So these are pretty significant results. My husband's here with my power cord because I forgot my power cord. Thank you. Okay, my whole family's here. Thank you for rescuing me. So this is the story of mercury in common loons. But common loons are not only exposed to contaminants in the environment, not only to mercury, there's a whole plethora of other environmental pollutants that common loons are exposed to. So this is work done by the Loon Preservation Committee and headed up by Tiffany Grade. And she's looking at not just mercury in common loons, but also dioxins, PFAS, PFOS, flame retardants, PCBs, organochlorine pesticides, as well as cyanobacterial toxins. Loons are subjected to a lot in their environment. And again, they're an amazing indicator of aquatic health and ecosystem health. And they're a wonderful model system for understanding how exposure to these environmental pollutants impacts a vertebrate's susceptibility to infectious disease. So this is a great work going on by LPC. And unfortunately, loons don't get the readings from lakes and ponds that you shouldn't be eating certain fish. And again, these environmental contaminants are local and they're long and they're persistent. So loons are subjected to relatively high levels. Okay, so finally, here we have loons being infected with malaria parasites for the first time. What is going on? Are we on the wave of emergence of an infectious disease in common loons? If we are seeing the emergence of malaria in common loons, because this is not a co-evolved, co-adapted host parasite system, again, we'd expect to see an increase in virulence, and we would expect to see disease and death. And this is exactly what is showing itself to us. So this is a female breeding common loon. She was found on Lake Umbagog in 2015. There were some campers that paddled out one morning in their canoe. They had just had their coffee, the water was nice and calm, and they saw something floating on the lake. They paddled up to it and they saw that it was a common loon. Fortunately, as there's a wildlife refuge there was a wildlife biologist who was then able to retrieve the bird. And through LPC volunteers, the bird was brought down into the hands of Inga Sidor, who's a veterinary pathologist who's been doing necropsies on loons for decades. She's necropsied hundreds and hundreds of loons. And it's really fortunate that this bird was found, she was freshly dead. So she had just just recently died. And Inga was able to cut her open, and she was relatively fresh. And this is what Inga saw. She saw something that she's never seen before upon opening the bird and doing her microscope analysis. So she saw a very large, enlarged, spongy heart. She saw degraded lung tissues, and she saw a grossly enlarged spleen. And these are signs of malaria parasite infection. But again, Inga was taken aback as she had never disease in the species. Again, if we are, we would expect, because this is a non-adapted host parasite system, we would expect to see increased virulence, including disease and death. And this is exactly what we are finding. So this is a breeding female from Lake Umbagog found in July 2015. There were a few campers that set off for a morning paddle after their cups of coffee. They were paddling on the still lake, and they found a loon floating dead. And she had just recently died. So because there's a national wildlife refuge there, they were able to retrieve this common loon, get it to the refuge biologist, who was then, through LPC volunteers, able to transfer her down to the hands of Inga Sidor, who is a veterinary pathologist who has been cutting open and necropsying loons for over 20 years. So she has looked at necropsied hundreds of common loons. This bird was in really good body condition. She was nice and plump, and she was a breeding female. But when Inga opened her up, she saw something that she had never seen before. First, she saw grossly enlarged spongy heart. She saw fluid-filled lungs. She also saw a grossly enlarged spleen. This made her think malaria, but she had never seen malaria in a common loon before. So this is something that she took further. So she did histopathology analysis, which is where you take small narrow cross-sections of various tissues, and you look at them under the light microscope. And what she found was that the brain capillaries of this breeding female loon were chock full of malaria parasites, so much that they were impeding or fully restricting blood flow. So she deemed cerebral paralysis as the cause of death from malaria. This is pretty amazing. This is what happens to humans infected with malaria. Oftentimes they get cerebral paralysis. And this is first documentation of malaria in a common loon. So this was a pretty significant finding. I was able to take tissue samples from various organs. Inga gave me tissue samples from various organs. I did my molecular work in the lab. I sequenced the parasite DNA, and I was able to identify that Plasmodium or malaria parasite to species. I know it's Plasmodium species Cat-testo-5 in this loon. This species we know is in native North American songbirds, but I also know from my work at the National Zoological Park, I was looking at malaria parasites spill over into naive zoo birds, and I found Cat-testo-5 had jumped into and killed Inca terns and tinamous and flamingos. So this parasite loves to spill over into new hosts, and it's able to cause disease and death as a result, because it's in a naive host that doesn't have the immune system functioning to beat the infection. Likewise, a recent study just came out from the Bronx Zoo in New York City, and this study showed that Plasmodium species Cat-testo-5 has jumped into zoo birds there, including penguins and eiders, and caused disease and death. So we know this malaria parasite loves to host switch, and it is pretty deadly. So we published our work in the Journal of Malaria, in the Journal of Wildlife Disease. And because loons are so well loved, it made a splash in various local and regional news outlets, including the Boston Globe and Audubon magazine. So, but again, if this is the emergence of malaria in common loons, we wouldn't expect just to find an isolated case of malaria. We'd expect to see this over time increase. And because we are able to retrieve common loons when they're found dead, and because we had heightened sense to retrieve loons, and if they're freshly dead, to bring them down to the the lab bench of INGA to do the necropsy and histopathology. We were able to find 5 more incidences of loon malaria in 2019. The lakes on which these birds were found are included here. Some of you may be familiar with these lakes. So all these birds were similarly impacted by their malaria parasite infection. They had grossly enlarged spleens, they had spongy enlarged hearts, and most importantly, their brain capillaries were chock full of malaria parasites. So like the Umbagog loon, they died from cerebral paralysis. Again, I was able to get tissue samples from these necropsy loons. I brought them into the lab. I did my genetic work, and I was able to identify them to species, and they were all the same species of malaria. This is species Cat Test 05, all in these dead loons. But the big question here is just how deadly is the emergence of malaria in common loons? We have just over a handful of common loons that were found relatively freshly dead that we were able to do the necropsy and histopathology analysis, and that I was able to do my genetic work on. So we have grouped up, joined forces to answer this question. So this is the team. This is Mark Pokras of Tufts University. He's a loon expert. This is Inga Sidor at UNH in Vermont, New Hampshire Veterinary Diagnostic Lab. This is John Cooley with LPC, and then Lucas Savoy at Biological Research Institute in Maine. This is down here, there is Eric Hansen in a canoe. He's from Vermont. He's a loon biologist for Vermont Center for Eco Studies, and also Ben Lundberg, who's a rehabber at Vermont Institute of Natural Science. All these folks have gotten their hands on dead loons and are interested in helping to answer this question. They also are on the receiving end of loons that have been found dead and frozen. So we are setting out, this is a project that's in action now. We're trying to answer the question, what is the full extent and threat of malaria in common loons? And again, this answer, pursuing this answer involves broad collaboration. So common loons are a model system, ideal model system for answering this question. Just as they're a great indicator of aquatic health, they're also a great indicator of the impact of emerging infectious disease in northerly distributed wildlife. So we know that loons are an icon of the northern wilderness. We know that this emerging infectious disease has moved north and it's impacting them. And common loons are often retrieved when they're dead. And that makes this the most ideal study species to look at disease emergence. So common loons are large-bodied, they're conspicuous, they're well-studied, and they're well-loved. So people know the loons on their lake. They're found when they die by recreationists as well as volunteers through nonprofits, as well as people who inhabit lakes. So we're able to find and recover over 100 dead loons in the Northeast every year. And these 100 dead loons in the Northeast every year have been recovered and stuck in freezers. Some of them, varying proportions, have been necropsied, not all of them. Tissue samples have been archived from organs from these birds over time. So in the middle here is a picture of Mark Pokras, one of my collaborators. He is a retired professor at Tufts University. He still actively does loon necropsy sessions with vet students every year. And so tissues are archived from each and every one of these loons that are necropsied and stuck in a freezer. And those freezer samples have been accumulating over time. So we have tissue samples from common loons that have been found dead in the Northeast and archived from the last 20 years. They're waiting in the freezer for us to do our molecular analysis to see if they have malaria. There's also samples from these loons. Some of these loons were found relatively freshly dead, and samples were taken to later be done, undergo histopathology, but not all of them have. So histopathology is where you do the cross-sectioning, staining, and light microscopy of various cross-sections of various organs. And this is how we can find if a loon has died from malaria by looking for cerebral paralysis. Unfortunately, the histopathology analysis is pretty pricey. It's $95 per sample. So a lot of samples are waiting to be processed, as well as won't be able to be processed without funds. So I'm going to be shameless here and promote this project, looking at the emergence of malaria and common loons with the collaborators I just listed, we are crowdsourcing for funds to do this work this summer on every dead loon that is retrieved in the Northeast by LPC, BRI, VCE, and the networks of volunteers that are out there monitoring these loons. We want to look for signs of malaria parasite infection, disease, and death in every loon. And the work is pricey. So we are hoping to raise $7,300. We're halfway there. If you're interested in supporting our project, you can check it. Get more information on our experiment.com page. The link is listed here. And we are halfway to our goal of $7,300. We have until July 15 to raise funds. It's an all or none funding campaign. So if we get there, the project is going to start August 1st. We're going to get our hands dirty. We are going to start doing our lab work. We're going to start doing our necropsy and histopathology work. If we don't get to our total funding goal, then we don't get any of the money. So we're hopeful we can get there and do this important work in common loons this summer. Again, this work is hugely collaborative. There's amazing work that goes on in the field, and the patience and hard work of loon biologists has allowed me to study malaria. And so I have so many people I want to thank. They're not all listed here, but especially the Loon Preservation Committee for supporting the project and letting me have access to so many samples, hundreds and hundreds of samples from live loons and also dead loons over time. And with that, I'll open it up for questions. I hope you're all still here. This is where I ended a little while ago and then nobody was there, but I'm happy to answer any questions if anybody has any.
Great, thank you, Ellen. We do have a couple questions in the chat. So I'll start off with this one, which is there an observed increase in malaria in humans up north? Or is this same phenomenon being seen in other species?
That's a really good question. So we're seeing it in birds. We're seeing parasites move north. In terms of humans, it's pretty tricky with climate change. We have some malaria endemic regions of the world that are actually drying out because of climate change. We have hotter, drier areas of the world. We also have wetter areas of the world. So we're seeing malaria increase in human populations in some areas. We're also seeing it decrease in humans and other areas. In terms of whether it's increasing or decreasing in other wildlife hosts, that question has not been pursued. We know malaria is really common in bats. It's really common in lizards. It's really common in deer. So my own work, we've found white-tailed deer to be infected, including up to 25% of white-tailed deer in the eastern United States. But in terms of how that's changing over time due to climate change is still unknown. These common loons are making a pretty ideal study system to answer that question, and we will be answering that question. We will be able to, especially through the work that the grad student Alyssa Newhouse is doing in the lab, because she's looking at work that she's screening blood samples from breeding common loons up to 2021. So stay tuned for our results. And then again, there aren't studies in other vertebrates other than humans, but mixed results for human prevalence over time due to climate impacts.
There's a question about the statistics that you presented at the beginning of the presentation. So those studies that were done on how loons are doing and the decline in the floater population. And the question is, what are your opinions on the status of loons? So in some states they're listed, in some states they're not. And do you think that, given what's going on, those listing statuses should be elevated, or just do you have any thoughts on that?
That's a good question. In terms of Wisconsin loons, where that study took place, they should definitely be listed. We should be paying attention to this. Unfortunately, those long-term studies are pretty difficult. The way our scientific funding works is that if you get a grant, it's typically for 3 to 5 years, and then your study's over. But Walter Piper was successfully funded by NSF for decades, and he was able to do that really important piece of work. And it's those long-term studies that are where we reveal those patterns of those floaters not coming back in We're not filling the spot of adult breeding loons that are dying. So it would be great if wildlife managers at the state level were to incorporate studies like that, but they're pretty much lacking for every other state. There's a nice study out of Canada from Birds Canada that's showing similar declines in common loons over time. And again, they have the long-term data that's through citizen science projects. But yes, ideally, in an ideal world, we would have these long-term studies funded by state and federal agencies. And we would be collecting these data and more closely monitoring common loons. There's so much we don't know, and so much that can only be revealed through these long-term studies. So, yeah, I think that species and the species committees that make these decisions need to be reading the scientific papers and taking in consideration what's going on in other parts of the breeding range of common loons, and also gathering information from folks like the folks at LPC and other nonprofits that are closely monitoring loons. So if I had it my way, they would be listed as endangered as well as so many other wildlife species. But yeah.
And are you aware of any loons outside of New England that have actually died from malaria, or has this only been seen in New England so far?
Good question. This has only been seen in New England. So Inga was the first one to see it, and but Inga's also an expert at necropsing loons. She's been doing it for so long, and she knows her stuff. So it's great that we have such an expert able to find that. I'm sure that common loon malaria mortality has happened, and it just hasn't been recognized. People don't have their eyes out for it. It's not something that's ever been seen before or that's commonly seen. So hopefully our work can stimulate more people to take that deeper look at a dead loon. And again, it has to be a relatively fresh dead loon. If the tissues have degraded at all, it's really hard to see the signs of malaria parasite infection, disease, and death. So again, we're fortunate here that our loon population and breeding loons on lakes and ponds in the Northeast are so well studied and so well monitored through LPC and other organizations. So we're really in a unique position to really better understand just what's going on with this emerging infectious disease in loons. So again, it's such an ideal model system, and we have so much to learn from our common loons.
There's a question about environmental toxins and climate change and mortality rates in loons, and which one of those things seems to be having more of an effect, or is that sort of unknown at this point?
This is all unknown. It's all, I mean, I'm a scientist. Science is amazing because you pick a slice of the pie and you go at it, and then so much is revealed. So again, loons are such an amazing model system, as they're such amazing icons, but also because they're great indicators of aquatic health and environmental health. So we're able to study the role of these environmental contaminants. But there aren't many other studies going on. And in terms of what's at play here, so that Walter Piper study, they just said they coughed it up to anthropogenic factors. And as you all know, probably better than I do, I'm not a loon biologist, I don't study loons, I study their parasites, but they face a number of anthropogenic factors. So lead ingestion, collision with boats, disturbance during the breeding season, predation, all those things are increasing over time. And in terms of what is their role emerging infectious disease in population decline is unknown. And again, we are in this really unique position to better understand that, because we're able to retrieve the dead loons, and we're able to look for signs of malaria parasite infection and disease. So we'll know more over time, especially the more loons that we look at.
Great. Well, thank you so much, Ellen. I think that's the end of our questions.
Great. Thanks for having me. I'm sorry about cutting out for 10 minutes. I don't know what happened. But
thank you for sticking with it and coming back and being willing to sort of rewind and and give your presentation again. I know a lot of folks were really interested in seeing, you know, the end and seeing your conclusions. So thank you so much. And I will direct everyone just one last time to the link that is in the description of the video here. And I've also posted that link in the chat. If you're interested in helping to fund the continuation of this work, this really important work for loons, please do check out the experiment.com page for this research.
Great. And if anybody has questions, feel free to contact me. I'm happy to answer more questions after the talk too.
Great.
And thank you everybody for being here. Happy loon watching.
Yes, everyone have a great night.